So the work by Anfinsen suggests that all of the information is in the primary structure, even if the protein's unfolded, to help lead the protein to its correctly folded form. But the question is, still, how does it get there? Is it a kinetically fast process? Is it a kinetically slow process? Does it occur slowly through steps? Does it occur all at once? What's going on? And so to think about this last element of the puzzle for Chapter 2, here's a bit of data that is typical for doing a protein denaturation experiment. And what one basically sees is that if you add a denaturant, whether it be temperature or whether it be concentration of a chemical, what one usually sees if you plot the fraction of unfolded protein as a function of added denaturant is that you see that it looks more or less folded in terms of its structure for quite a while, and then all of a sudden, kind of like when you're titrating a strong acid with a strong base, you see a very, I don't want to say rapid, but you see a change that occurs over a very small amount of added denaturant that kicks it from completely folded to it's essentially weakly or completely unfolded. Which sort of suggests, with this sharp transition, like in a titration, that folding is all or nothing. So what we want to consider now is it really all or nothing, and how fast does that take place? So the first thing we're going to do is to think about folding times, and this is going to be a little bit of just fun math. So let's suppose for a second that folding isn't really happening as an all or nothing step, but it's doing it sort of through a random search of conformations. So let's just say we have an amino acid or a protein that's made of 100 amino acids, kind of like ribonuclease. And let's just assume, for the sake of very simplistic ease, that each amino acid can search out 3 possible conformations, which really isn't a lot. It's just saying maybe the amino acid can go in the X direction or the Y direction or the Z direction, and it could actually do a lot more crazy combinations beyond that. But if we mathematically consider what that looks like, the total possible conformations is staggering. It's going to be 3 to the 100th power. That is a number that I can't even kind of comprehend. But let's do better than that. Let's assume that each conformational switch, as it's doing its search, is fast, on the order of 10 to the minus 13 seconds. That's pretty flippin' fast. That's really fast. To figure out how long it would take for the protein to randomly find its way into the folded conformation at 10 to the minus 13 seconds, that's at the rate of diffusion, guys. That would simply be the product of these two values. 3 to the 100th times 10 to the minus 13 seconds per switch would be around 10 to the 35 seconds. Now, a little bit of quick math will tell you that that's older than the age of the universe, or really, 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 really slow. But here's the thing. Folding times of proteins, even in the Anfinsen experiment, seconds, minutes, hours when you're doing it in solution without any help. Seconds or minutes compared to the age of the universe. So clearly, the process is happening faster than that. And the question is, how does it do that? So, there's a paradox here. It should take longer than God's creation, but it happens a lot faster. So, a couple of hypotheses for how this happens. And I'm not really interested in having you know every painful detail, but the big picture. So there's 2 methods that have been proposed in the big picture. First is what's referred to as progressive stabilization. So in this method of folding, we don't think of it as occurring by a random search, but more like the way you would think of a Rube Goldberg machine. So for those of you that don't know Rube Goldberg machines, there's sort of one process that starts something into motion, and through a series of complicated steps, it does some final job. In this case, our final job is to end up at the native conformation. But the way the model looks is kind of like this. So this, again, is a cartoon out of your book. You start in some sort of denatured, unfolded state. Maybe there's a little bit of residual structure, maybe there's not. But the idea in progressive stabilization is that it doesn't really just happen in a truly random process, but you get, say, one region of the protein that does maybe a random search or a pseudo-random search, but in that pseudo-random search, that starts to lead to a little bit of correct organization for the final protein, which sort of gets parts of the molecule in the right place. Well, now then, for the remaining regions of the protein, it can sort of take little molecular cues off of the first part that's folded to help it not go through a random search, but through intermolecular forces, whether it be the hydrophobic effect, whether it be through hydrogen bonds that hook onto different regions of the protein. That helps the system to drive itself towards a conclusion faster than every single thing doing a completely random process. And each step helps to facilitate the folding of the next step until you ultimately end up at the native conformation. So in a pure aqueous solution, like with antisense ribonuclease A, this probably is going to take a little while, maybe a few hours. But we know that in biological systems, this can happen a little bit faster, i.e. seconds to minutes. So in this case, maybe it's not working completely by progressive stabilization, but maybe the cell is doing a form of intervention. That is, it's not doing it all by itself, but maybe the biological system sort of rigs the system a little bit, puts enzymes in place that help to maybe provide some extra free energy from enzymatic reactions that help to fold it. Or maybe you use what are cutely named chaperone proteins, sounding like what you think it means. A protein that sort of serves as a template to make sure things do what they're supposed to do. They chaperone the folding process such that it works. Some of you may have even seen some of these names in other courses. So one important chaperone that shows up is called HSP 50, HSP 70 is the one you usually see in books. But other biomolecules that sort of, point things in the right direction. Maybe they act as a template. Maybe they provide some enzymatic process to help drive the process forward. So this is just an example of a model that was taken out of another textbook, out of the Linenger Principles of Biochemistry, one of the other two major biochemistry books that's used in the universe. And this is just showing how enzymes can do this particular process, or facilitate the process. And I don't need you to know the details here in any kind of memory intensive way, but the main theme here is that you might have some sort of unfolded protein that is working together with an enzyme that undergoes some sort of that helps to facilitate some sort of catalysis that might burn an ATP molecule in the process. But the idea is that these enzymes, DNAJ and DNAK, will, through a free energy process, will then bind an unfolded protein, and through a series of steps, the binding of this protein can then ultimately lead to a set of processes that helps to kind of work the folding out in a facilitated manner. So again, in biological or cellular systems, this can a lot of times be an assisted process that helps to speed the process up from hours to the order of seconds to minutes. So that's one method. The other method looks at life very differently. And I'm not going to say that the progressive stabilization is right or the second method's right. It's probably two sides of the same coin looking at it from a slightly different point of view. But in the second method, they don't say, nah, it's not working from this sort of Rube Goldberg, this step leads to this step. In the second sort of scenario, and again, these are probably sort of two sides of the same coin, it's more of a free energy driving force. So in this particular process, you're not saying this step leads to this step. You're just saying if you have an unfolded protein, it's free energy uphill compared to the final folded structures down at the bottom of the hill. And if you start to fold the protein in any way, shape, or form that's correct in conformation, it's going to start to fall down the thermodynamic hill. Rather than go up to some other terrible state, it's going to fall down the hill. And it's probably going to fall down to what a lot of people term sort of an intermediate form of a stabilized protein called the Motin globule, which is kind of like getting it close but not fine-tuned. But once it quickly falls down here, then it's going to, again, sample some thermodynamically distinctive conformations, but eventually it's going to keep falling down the hill until you end up in conformational states that are the low energy states that are much, much more favorable and stable than the thing that you started out as. So in this worldview, the idea is that you're falling down a thermodynamic hill, and it's not so much that there's a step-by-step process, but it's driven by free energy. So it's sometimes called the free energy funnel. And like I said, these methods, neither of them are probably perfectly right, and they're probably both working together, and there's probably even a third or fourth method that could more accurately depict it. But in the big context of how this process works, it's probably a combination of these different factors. So that more or less wraps us up with Chapter 2, and the only thing I have left is to make just one little quick add that we didn't get to earlier, or we'll kind of restate one little hanger-offer topic that doesn't really fit with any of this stuff, and that is, so far we've been talking all about folded proteins and unfolded proteins that are in the wild type states, but like I was telling you guys when we first did the amino acid tour, it turns out that you can do post-translational modifications that can affect the game of how these things fold and work as well. So we already mentioned the hydroxyproline once upon a time. Again, don't need to memorize it. But again, the idea is that sometimes when you work with proteins, or when nature is utilizing proteins in a particular way, there's going to be groups that get added or subtracted, like carboxylate type groups or like phosphoryl groups, that their mere addition or subtraction will create a new important electrostatic or take away an important electrostatic group, or maybe it's a bulky group or what have you, that will immediately lead to changes in three-dimensional structure that affect protein function. And on that note, I think we'll wrap it up, and I'll just say we'll see you at our next webcast.